Prophet Gill when during my time there. It was very helpful for me in my career. But he has a connection to this piece. So you know how like my sort of pride / chagrin that I was here 37 years ago, and I was kind of beating everyone in the room. But then there was a guy who came into the library the other day who was here in '75, which is just kind of after the station started. But Don was here in 1968, which is before the station was here. And Don was on the original dive team that came out to assess this for UBC, whether this would be a good marine station site. So Don was possibly partially responsible for the fact that the station is here. Okay. So, in a very, very small way. There you go, why not? Don did a bunch of work on behavioral ecology and birds and stuff, and you can have a chance to chat with him later because right after this we're going to go to the director's house. We have permission to go down there and have a meet and greet happy hour, and we'll hope you join us, BYOB. Okay? So right after this we're going to go down to the director's house. Okay, now for the main event, we wanted, for our class, which is called Rapid Evolution Stickleback, we wanted to bring in the person who's more about stickleback than anybody else in the world. And I think that's pretty much true. Half of it might be wrong because we argue about it, but no, Tom is really like the world's expert at this point on stickleback. And so for stickleback researchers, but also just for people who want to understand how organisms adapt to their environment, so rapid evolution, and just to sort of understand the complexity of nature and adaptation, Tom is a great speaker to have out here. So Tom is from the University of Victoria, where he's worked for many years, and he's done most of his research in Haida Gwaii, which you will hear all about. And you know what? I think you should just give your talk now, Tom. I don't need to say it. So thank you, Tom.
Bigger than some of my classes, actually. So, thanks for the invite, Sarah. Thank you very much, and Don for co-driving on the way out. I didn't want to drive all the way by myself, and I don't trust the autonomous mode on the Tesla. So. All right, Sarah asked, or if I could sort of focus on my stay at Grizzle Lake, where I've done sort of some 25 years, and we lived quite a long time at the lake. And that allowed certain insights that probably wouldn't have happened with the more conventional approach to research. And what I want to do is sort of take you through, first, my perspective here on what the significance of interpopulation vary is. Basically, here's your standard adaptive landscape. Everybody knows this for the last 50 years. This is the frequency distribution, etc. And this is by large considered a fitness landscape. So high fitness basically dropping down to adaptive valley, etc., back up, etc. So that's what I want to evaluate, etc. Secondly, I want to look at interpopulation variation with the, sort of the same type of theme of like what does it mean for this versus that peak, etc. That's going to be a shorter section, and finally a very short section will be on transplant experiments, where I try to determine sort of whether after you introduce something into an ecologically opposite habitat, would it evolve in the direction that you predict, and maybe, you know, in my lifetime would I be able to detect any difference at all, etc. Okay, so a little first of basic natural history, etc. As you all know, threespine stickleback, belly bone, northern hemisphere, oceanic waters, coastal, estuary streams and lakes. And actually it's found all the way to the middle of the Pacific, because steelhead have been captured in the middle of the Pacific, full of stickleback. So we know they're very widespread oceanically. Studied for a long time. Basically, we had very quickly here, obviously, oceanic stickleback migrate to coastal waters, estuaries, freshwater. The male establishes territory, builds a nest, courts the female, and then aerates the fertilized eggs. Pretty standard stuff. And then basically after he's finished, usually 2 weeks or 3 weeks, he goes belly up, and basically the fry migrate to the ocean. So anyway, that's the typical background to oceanic fish. But it turns out that right around the globe, these oceanic estuarine populations give rise to freshwater populations, out of which some of the fry are a little more tolerant of freshwater and move in. So we've got sort of the well-established, well-known origin of these freshwater populations. So what do we know generally is that the freshwater populations tend to be smaller than the oceanic ones. They have relatively few bony plates that protect the fish during predator attack. We know from not my work, other people's work, that there's high heritability in most of these traits that differentiate the oceanic and the freshwater population. So this is a typical marine form and a freshwater form. Okay, so that's a bit of background. So most of you know where Haida Gwaii is, etc. And after I did my degree in England, I headed out west, etc. And so there was an old log cabin built by a settler, British settler in 1914 or 1913 at this little remote lake. And beautiful little cabin, about 100 plus years old. But he went back and fought for the king and he never came back. And so this cabin was basically abandoned for some 60 years. So anyway, most of the time we hike in. It's a 2-kilometer hike, but a lot of the equipment, you just get a float plane and take in the big stuff, etc. All right, so this is a little cabin. It's sort of just like this building, etc. Obviously, it's pretty rustic, etc. And it looks like that for many months. But that's what it looks like in winter, etc. Not every winter, but many winters. And from this old cabin, sort of, we basically lived year-round in this from 75 to 85, not just summer, but year-round. And then 86 to 90, I was teaching at University of Alberta. We came back for your typical summer research, basically from May to August. And then since 1991, we've been only going for pretty well July visits. So here's me when I was a real biologist, etc. Sort of working on a microscope in the old cabin. In about 1982, I got an Osborne computer, and who would ever need anything more? A little floppy disk that held 90 KB, and went, oh, unbelievable. Anyway, at that time, that was phenomenal. And so we powered it all with solar panels, etc. And I also used shakes, built a windmill with shakes, put a generator on in winter, we charged the batteries because we didn't get too much sun in winter, etc. So anyway, that kept lights going, computer going, sort of a nice printer, etc. Okay, so that's the background. So this is the lake, etc. And basically, it's very interesting in the sense that it has what's referred to as a giant stickleback, typically modal size around 80 millimeters, etc. The lake actually looks as black as that in the real world. It's a dystrophic lake. It's a high, tremendous amount of tannin, etc. So I was able to do, after 10 years, you can accomplish a reasonable amount. The population of juvenile stickleback was about 10 million, based upon the number of nests and egg counts. Sub-adult stickleback, based upon mark release recapture, about 500,000, and sort of adult stickleback were about 80,000 from mark release. So I had a good idea of what type of sort of demographic situation I was dealing with. So the age structure was also quite interesting. And unlike most stickleback in most populations, actually they live for up to 10 years. They're iteroparous. On the stickleback everywhere are several pairs, like a salmon, belly up, and that's it. These are basically their pairs. They breed in their third year and continue to breed. Even the 8-year-olds I found, the females are still gravid. So they continue. And it has something to do with the black water, the highly acidic waters that we still don't quite understand. So what I want to know from it, basically, is there spatial and temporal variability in the stickleback? I'm interested in variability, so that was one of the things I was going to do. So I did 3-dimensional sampling of the lake, so at 96 sites, and I sort of marked these with sort of little dots, basically, not only sort of horizontally, but I did from the shoreline to the middle and from the surface down to the bottom, trying to get a full, obviously, perspective of how much variability will you find in the lake. And I was primarily focused on the defensive armor. I also did a little bit on trophic structures, but the armor was on the issues. So this is sort of the lake, etc., on a nice summer day. And so, obviously, as a field ecologist, you want to know who eats these things, what are the predators, etc. Where do they forage on the lake? Pretty basic stuff. What size classes of stickleback to be? This is so basic that every field biologist would want to get this information. How much do they eat? How much does each predator eat, etc. And so to accomplish this, I did basically weekly, bi-weekly transits, basically of the lake, of all the predators that you can see, that basically dawn to dusk multiple times a day. So, okay, that's just sort of a bit of natural history. At the time when I started, stickleback workers, including ourselves, really thought there was 2 things that were happening. Stickleback had a lot of predators, in other words, predatory fish, or they had nothing, etc. And so I started out, I knew there was some trout in there, I didn't know much. But then, week after week, you gradually find out that, turns out that there's a substantially greater diversity of predators than I initially anticipated. Not that you would see on any day, but when you're there weekly throughout the year for multiple years. So obviously you can see the kingfisher, the hooded gans, the red-throated loon, the otter, basically Pacific loon, cormorants, common loon, common merganser, redneck grebe. All of these I was able to get some information on, etc. And so what was important was basically where did they forage? And so what you can find out, basically is arranged these from the top left, which is most of the littoral, and obviously a kingfisher is hunting near shore. And then down the left-hand column, basically to the cut trout, which was primarily littoral. And by littoral here in these blackwater lakes, light doesn't extend beyond 2 meters. And so there's no more basically primary production, so to speak, there's no more new farm. And so normally 2 meters is sort of what defines the littoral zone. And then basically Pacific loon all the way down to the redneck grebe, which basically foraged everywhere. So, well, obviously with that information of what species there were, there's actually 23 species of avian piscivores. Basically, sort of trying to figure out basically how many they eat. And I did that in multiple different methods, etc. So here's the list of these, and the ranking of them combined with the total number, and to determine the total number of stickleback to trout ate, I captured trout every week, basically looked at the body size, looked how many they ate, basically I went year after year, month after month, etc. And so this is the type of data you get that should be basic to almost any study of natural population. So you can see cutthroat trout, 300,000, and the light and the dark indicates basically different size classes. You can see most 10 to 40, these are 1-year-old or less, etc. And then the second is the common loon, dolly varden, old squaw, etc. Sorry, long-tailed duck at that time, that was the name. So anyway, and down to the bottom were obviously echinodioids, and I didn't really know how many there were, but I didn't think it was very much. So now I have an idea of where they forage, which ones are the most important. So every day we're now going to get a little bit more information on these, etc. So here's just an example of the redneck grebe. I really like them because they bring all their fish to the surface, etc. And they spend a lot of time with sticklebackers, put up their spines, etc. And they can't be swallowed like that. And the predators got to basically break them down or break the spines. And so So normally it was 4 minutes, 5 minutes for any capture before they would swallow it, but actually up to 18 minutes. That's how long before they finally swallow it. And many of these actually get away, and you find them in the lake. They have these beautiful, what I call ibis scars, right? They've been attacked by a bird, etc., manipulated the scars there, but they've gone away and they've survived. So, Vermeer argued that there's information in these individuals that basically get away. And he argued that these are the individuals that have been tested by natural selection and probably have higher fitness than those that you find that don't have any marks, that have never been attacked at all. So he tried to focus attention on when you're sampling and analyzing. This is a very important insight in terms of variability in natural population. So I did a whole series of experiments. These are some of the tanks that flew in with the plane, etc. And you can see the water is very dark, like it's very, very stained, and so you can't see down into the aquarium, you have to see through it. There's just a photo of a cutthroat trout in the tank, etc. And I don't think I could do this now legally, but I offered them. And then I eventually, some of those got away, and I kept those, and I watched how long they survived, and I eventually sacrificed them and looked at all the injuries that these fish that got away from the trout, right? And you can see it in the tanks, the trout grabbing. And then, let's go a second. So yeah, I was interested in sort of what the consequence of a toothed predator was compared to a compression predator, etc. So here's just all this stuff is published. I'm not going to bother you with the publications other than maybe just a few. But here's just sort of basically fish that have relatively few bony plates, right, have high levels of injuries. In other words, the plates protect the skin from puncturing from the predator, tooth predator. And the more plates you have, in fact, the fewer punctures. Well, the lower one, which is more important, is how long do they survive, etc. And the ones, in fact, that have higher punctures, surprise, surprise, have relatively low survival. And therefore, in the wild, if an individual that has few plates does get away, sort of not good prospects for that. But if you have a lot of plates, right, you have a good chance of survival. Okay, so that was sort of that first part. And out of which I eventually discovered that these plates differ in their functionality. About 4 or 5 of them function to buttress the spine, because the predator is trying to break the spine this way. And these bony plates basically disperse the forces, right, that happens when the predator is directing this way. So that was a sort of an identification of functionality from this. And then I found that these anterior plates are important to resist puncturing of the lateral line nerve that came up. So this was published a long time ago. So quickly now to spatial and temporal variability in number of plates. So here's a distribution of plates basically in the population. It runs from, you can see, on average 3 to 7, etc. So in the mode, basically, you can see 4 or 5. So there's that variance from that first curve I showed you, right? So you've got this optimal form up here, and then it's spread. So when you plotted actually the distribution from those 96 transects, and all I've done is now take this relatively crude axes, etc., project it down from the lake, where in fact the sample was taken, and said, what did the fish look like at each of those positions? And so this is the best that I could do that corresponds to the shape of the lake. And so what you can see here is the y-axis here, vertical axis, is in fact numbers of bony plates. So the fish near the shore, etc., this side, right, have elevated levels of bony plates. Right? No surprise, this is where the cutthroat trout are primarily foraging, etc. And what do we have in the middle of the lake? Well, there's a dip. So the fish have fewer lateral plates in the middle. Well, you only have to go back and see that it's the majority of dying avian piscivores that are in the middle of the lake. And so I thought this was pretty interesting because that curve, that basically was not simply just one optimal mode, etc. And so when I did this over 3 years, there was temporal variability. So this is the same month of the following year, and this is, you can see these are not small samples, these are big samples. What you still get the dip in the middle, but it varies, etc. So temporal variability, spatial variability, etc. And so anyway, I would thought that that's pretty well all you need to do to show adaptive variability. And what I did basically is I looked at the proportion of basically trout to birds, etc. Because I had all those numbers, month by month, year by year. And this is basically a plot, right, of the relative importance of primarily trout predator and no birds, versus down here is primarily bird predator, and no trout and bird predators. And so what you can see here, basically, is this goes up and down and up and down and up. So over time, the proportion of the sources of mortality vary. So this here, in fact, is the distribution when you actually look at the bony plates of stickleback over this highly oscillating selective predator pressure. There's virtually no response in the adult fish. Right? Oh, it's not a flat line, but there was no significant relationship between more trout predation or more bird predation. And I thought, well, that's sort of, I didn't really expect that. But if you think of the survivorship curve, right, where is most of the mortality occurring? It's not on this flat line from age 3 to 9, right? That's basically, right, your standard J-shaped survival group. It's here. So I started to look at all the subadults. And this is what you get for the subadults. You get this striking coupling between more trout predation, greater prevalence of high numbers of plates, more bird predation, and drops down. And so when you actually do a multiple regression or whatever, it's actually the partial correlation for this, almost 70% here of the variation in the lateral flights can be accounted for by the difference in number of trout versus bird predation. So that to me was sort of an interesting component here that I was aiming here. So obviously if I was to look at fitness, it wasn't going to be one at the top. It was going to obviously be a little one, and then maybe dropping off a bit. So that was sort of my first sort of insight there that this adaptive landscape just didn't fit basically from the preconceived notions. So when you're watching predators a lot, you can see they are basically they die down, and sometimes they come up and they've got nothing, etc. Sometimes you can actually see them from the swirl in the water that they're chasing something, but they still come up with nothing, etc. And sometimes they come up with something. So I was able to do this for several different piscivores, avian piscivores. So for the common loon, which was the second most abundant predator, etc. This is what you get. So obviously there's individuals that escape, and I have no idea how many that is, right? But basically, there's 55,000 that escape during pursuit, because I know how many in the year that they eat 100,000 fish, and I know the percentage efficiency for these. And basically, 770 escape after capture. Okay, out of which 804,000. Okay, so I didn't know if this is really meaningful, right? So I thought, okay, obviously adaptations are occurred during the pursuit. A fish swims there, away he goes, or it gets captured, and then what's the outcome? You can get away, or it can be eaten. So I also did basically this for redneck grebe. And what we got for redneck grebe with the same type of sort of approaches, etc. You have escape, 29,000 escape, only about a half, basically. They're not eating as many. So that's sort of a pretty straightforward thing. So whatever type of selective pressure is facilitating basically variation that deals with pursuit escape, say whatever. Obviously you expect the common loon to have almost twice as much importance. But when we look at the redneck grebe in terms of post-capture, 1,600 escape, right? And I thought, well, that's sort of interesting. It's a slightly smaller predator. It's not as efficient, right? A common loon's bill is this big, and they have an easier time. But redneck grebe is like that. So more get away from the redneck grebe, okay? And so what you have here, you've got 100,000 eaten by the common loon, only 8,500 eaten by the red-necked grebe. But when you look at the numbers of those that escape after capture, only 770 escaped from the common loon, but 1,600 escaped from the red-necked grebe. Now, that's sort of pretty easy, obviously, to see the implications of that. But an uncommon inefficient predator has potentially more of an effect on selection in this population than the more common predator. Right? Just focusing on Vermeer's idea that those that get away have been tested. Right? And so this was a useful bit of insight. I mean, it's just not a bird predator. There are bird predators and there are bird predators, etc. And so something that you would maybe initially dismiss as being relatively minor with regard to the demography of capture, etc., turns out to be potential. Okay, so I don't know the functionality of this difference, but the reality is if you were to really develop this, this is the type of things that you want to sort of compare. And what is that post-capture sort of attributes of the common loon versus the red-necked grebe? Another aspect of that is basically when you look at the number of bony plates, not only the numbers important, but each of them has positions, like I told you. 1, 2, 3 are good for protecting the skin, and 4, 5, 6. And so you can actually find many different phenotypes. So this particular phenotype here, this is just a frequency, numbers of individuals. This phenotype has plates, 5 plates, 4, 5, 6, and 7, right? So 4 to 7. So there's also these other phenotypes that have 3 to 7. They have an additional, right? 4 to 8. They have a plate a little further back, right, etc. So whatever. I don't want to label on these, etc. Labor on these. But when you look at the frequencies of those, this is what you get. And obviously, pretty standard. That's what your bell-shaped curve looks like. You've got a modal form, etc. Progressively less common of these more peripheral phenotypes. Okay, so you basically that's what that frequency diagram. So I thought, okay, what does this deal with regard to predation, etc. So I looked basically at sort of how many of each of these phenotypes were injured, paying attention to the idea that escaped fish with injuries indicate they've been tested. Etc. So this is the data that you get. So what you find here is that the modal phenotype, the next to the modal here, has big, exceptionally low numbers of escapes. And as you move to the more peripheral, uncommon phenotypes, the probability of capture with injuries goes up. Now, we sort of know this at the edge of the curve that gets whacked all the time. You know? I mean, you can't swim properly, whatever it might be. So this is really consistent with it. But keeping in mind Vermeer's idea, right? Does this actually mean these individuals have a higher fitness, these incredibly rare phenotypes? If they do, why aren't they more common? Right? That would be the logical thing. So basically what we were able to do, because these fish are long-lived, I was able to age many of them, and I could establish a birth year. I would establish each cohort. And so I could go through all these fish and age them, basically, and separate multiple cohorts and follow the frequency of these different phenotypes within a cohort. Right? Not different cohorts, but within a cohort. And so we did this, actually. So this is the fitness of the peripheral phenotypes, all in relation to the common one, to the first one. Okay, so what do we expect on average, right? Thinking conventionally, here's the fitness, here's the fitness of the modal one, 0, everything is relative to that. So all these sort of hopeful monsters at the edge, you'd expect the fitness to be less than the modal phenotype, okay? So this is where sexes combine, and basically this is what you get. So these are the different phenotypes. So there are those that are clearly at a disadvantage, but here's all the others. The majority of them, even the most peripheral one, have a higher fitness than the common mode. So this is real puzzling because, right? Why aren't they more common? right? That's what you'd expect. So we split the sexes on this. We split the sexes, and what you get, maybe same way, both sexes show, in fact, elevated fitness for all these peripheral phenotypes relative to the modal phenotype. All right, you put that together, you know, with this bell-shaped fitness curve. Clearly, this doesn't make sense at all, that this represents fitness. Because these individuals have elevated fitness relative to the mode. So there's still a few that are below. Okay, so we published this quite a few years back, etc., in evolution. So, okay, another aspect of this is bilateral symmetry. Okay, so I've talked only about the phenotype based upon the numbers on the left side. But when you look down at the fish and you score these bony plates on both sides, a lot are symmetrical, but a few are a little off on one side. And so here's an individual looking down. Basically, this is the left side. It's got one additional plate, number 3, that's not present on the right side. So this is bilateral asymmetry. Right? It's present in almost all organisms, etc. Okay, so in case you aren't familiar with the literature, basically subtle asymmetries basically are thought to represent developmental noise, basically phenotypic noise, and often are thought to be costly to fitness, not necessarily neutral or costly. So that's the tremendous rich literature supporting that. So anyway, I looked at this issue with regard to these stickleback, etc. So typically, most literature, you subtract right side to left side, you obviously get something negative, positive. But the tendency is to use the absolute value as a direct measure of how many of these fish are symmetric or asymmetric. And so I did this. So what would you expect? Well, if it's developmental instability and phenotypic noise on these nice natural populations, etc., you would expect it to be relatively uncommon. Asymmetry should not be common. Right? Maybe in a disturbed population, inbred, whatever, you expect all sorts of things. But these are natural populations. So this is, we did this for some 60 plus populations, we're now over 110. And this is the frequency of the absolute asymmetry. For each of these bars is a different population. And you can see the frequency of these subtle asymmetries goes from basically 25% to 65%. There's a couple down here that are fully plighted that don't do not asymmetric. So clearly, it's not consistent with observation, right? You would have expected relatively few. So what basically we decided to do here, what you look at this is that there's pretty well similar proportions of right side and left side asymmetry. You're just as likely to have an additional plate here or here, etc. So that's what produces what they refer to FA, so the mean of 0 for the population. All right. So I wanted to look at the fitness. Why not? I had all these cohort data, etc. And I looked at the fitness of the asymmetric phenotype relative to symmetric. So we predict low fitness, etc. In this particular example, so that is the fish has an additional plate on the left, right? So what did we find here? So that's the asymmetric, etc. So left bias asymmetrics, again, we expect everything on average to be below the line because this is developmental instability. This is phenotypic noise. And what do we find? And these are different years. These are different cohorts. They all have a higher fitness than symmetric. Completely counterintuitive from this vast body of literature. I don't have an expert around here, but there's a rich literature that would support the fitness of these asymmetric should be below the line, not above the line. So here's another one. Here's the right bias asymmetric, basically one additional plate on the right side. So maybe there'll be a difference there. Same thing. Right and left asymmetrics have higher fitness, evolutionary fitness than symmetric. Why aren't they more common? Well, they are not common. On average, 40 to 45% across the population. So why should this be? Right? I mean, There's no magic here, you know, these are ordinary populations interbreeding big populations. How can you generate this type of, I mean, these up here are basically numbers, so this isn't, these are not 5 to 10 or 15 individuals, these are big samples. Anyway, so because I'm a functionalist, I thought, what have we got here? So some of you are probably familiar with this literature. It's often thought to be hemispheric dominance, right? Our left brain and right brain are not the same, etc. How many are right-handed here? 90% of you will be not right-handed, as am I. So this is absolutely sort of the norm, etc. So that's what's referred to as laterality. So taxonomic widespread for preferential use of the left and right based on perception or action. So this is right, etc. Some birds use the left eye for looking at predators. That's laterality. So here's Aaron, right? She's a lefty, right? If you use Game of Thrones, or is that too old for? Okay, Aaron that are dealing with hand-hand sport have a much higher incidence of left-handedness than right-handedness. Right? Does anything ring any bells of why that should be? Why, as a combat individual, a lefty would be more?
Because most people fence right-handed. Since most people fence right-handed, most fencers are used to fighting a right-handed person. So a left-handed person,
absolutely.
Most people are less experienced in fighting them.
Yeah, it's lack of predictability. That's what gives them an advantage. That's the typical interpretation of why the great swordsmen are left-handed. And as a bit of an aside, they're also more likely to be president of the United States. It goes on, tend to be smarter than us righties, so whatever. So whatever, there's a difference between left and right. So obviously, there's a difference in the, not the size of the brain, but the actual prevalence. You have different sensory modalities, and you've got a part of the brain that is a little more efficient at doing this, and another part of the brain that's doing something in an opposite way. Here's the basically humpback. When you take a look at humpback, it's not my work, etc. Much of the scarring is on the right side. So when they're actually breaching, they're breaching in the same direction, etc. When they go to the bottom, they tend to rub against the bottom with the same. So this is widespread, and even Paleozoic trilobites show this asymmetry. So here's a common sort of retro-to-do. Basically, it's got an ocean fish back, and you can see the head of the fish is to this side. So anyway, the bird might be manipulating, but you often find basically the belly of the fish is facing in, which usually means the bird, I got it from below. That's the first thing. But the head is to the right. So if you look through the geometry, you can sort of figure out, so why would the head be to the right? So there's some indication of laterality. Here's a red throat bringing a different fish, some of the sort of sand lions type, and it very consistently had the head in the opposite direction. So it could be that these fish are evading in different ways, or it could be that the predator is attacking these different species of fish in different directions. So here's my stickleback. Obviously, that was a preamble, etc. Staring at us. This is one that has a right pelvic fracture, and this is the one that has a left pelvic fracture. Very easy to score. So I spent quite a bit of time scoring this in relation to the habitat. So what did I find? And so what we have here, you can see here, above maybe that 0 line, there's the right bias fracture and the left bias fracture. And here's different years of sampling, etc. So these are all these adults that I captured. So what do you get? Well, that's sort of interesting. The majority of years, you can see, is fish that are the pelvis is broken on the left. But those pelvic spines are completely identical to each other. So it's not that one spine is weaker, right? There's something about how predators have attacked this, or how stickleback may be evading in one direction that facilitates, in fact, this 5 years ago. But there was one year, basically 1983, which was all right, not all right bias, but a shift to right bias. It happened in both sexes, etc. So that indicates that not some underlying feature, but maybe temporal variation in how predators capture or handle these. That was the inkling here. So I also, because I ate these fish, etc., when attacked, basically, so little fish, to the top of the survivorship curve you got into. Now you might not expect anything different here, but this is what this looks like. Overwhelmingly, when you're little, you're more likely to get whacked on the left side, or you're more likely to end up with a left pelvic injury. And as you get older, these are all adult fish, by the way, that I've captured, but because I can tell the length of growth, I can back calculate an age when the fish was captured. And so what you can see here is that the fish that have been captured more recently, in other words, the adult fish, basically are symmetrical, equal proportion. Which is at 2017 to 19, I'm paying no attention to this fact that it's higher than that. Okay, so that's sort of interesting. So I don't really understand yet what's going on, but these are the two plates that basically wobble back and forth between left side. This one is relevant to whether the pelvic is really strong. This one is not. So this is an incomplete part of the paper that I'm not sure I will be able to resolve. But nevertheless, coming back to the original here I've got this tremendous variation in this adaptive landscape. And the edge of that little mountain here is filled with individuals that have the same fitness as those that are in the middle. It is not a fitness landscape. So this is sort of quite an important issue of how we approach populations, how we study them. Okay, so the next thing I'll do real quickly is interpopulation variation. I sampled basically a lot of lakes, etc. 700 about in total, 140 stickleback, mostly in habitats, no native, non-native fish species. We know the lakes are about 10,000 because Haida Gwaii was glaciated. There was possibly a refugial area in the Hecate Strait, but all these lakes themselves didn't exist 10,000 years ago. So we know basically the fish that colonized them basically have evolved basically over that time frame. This is some nice pictures here. This is the type of habitat we went to all these different lakes, etc. And so you end up with a lot of variability in morphology, which we know are heritable traits. And pay no attention to this, but when you do principal components of all the morphology, you find, the west side has got more armored fish, etc., etc. So I don't want to dwell on this. And so this comes to my favorite person, etc. Right? This is, of course, Stephen Jay Gould. Stephen Jay Gould. He was the most forceful thinker in North America evolutionary thinking. And he's written so many books. Unbelievable that. But this is what he said back in '84, quite a time ago. It's time for the pendulum to swing back to a position at the pluralistic middle. I believe that both the great suprematists of the 1930s and the great synthesis were correct. Some geographic variation within a species is clearly adaptive, but much is a non-adaptive product of history. Well, as much as I like this man, it's the complete opposite to his conclusion. And so this is just sort of a summary of the actual morphology. The sense morphology was unbelievably predictable based upon the spectral characteristics of the lake, because that set in motion the type of predators that lake would accommodate. So stained lakes had a certain assembly, etc. Okay, quickly, the last thing I'm going to do is a little bit on transplant experiments, which I think is the most pertinent current. So this is the type of fish that you get in large lakes. Here's the Mare Lake and Drizzle Lake, Susan. These are what you get in small lakes. You have reduction in armor, small spines, etc., etc. So what basically I wanted to know right? Because I knew this for about 10,000 years. So if I transplanted something like this into a small pond, right, how would there be any changes at all, right, in a pond that had no predators, was basically ecologically opposite? So what do we have here? So what I did, I undertook 4 transplant experiments, basically. 3 from Bear Lake, which is this big lake here, which is these giant fish, and I transplanted them into 3 different ponds, that were basically as different as could get. Trophic, different predator regime, etc. Water chemistry, very, very different. Clear water versus dystrophic water. And so just a few attributes of morphology concern. We got a sample from 1929, which I didn't do, etc. So area 490, dystrophic, stickleback diet, etc. So that's that attribute of a giant population. And this is what I introduced it, just a few kilometers away. So this little pond here, no fish, no outlet, 0.05 hectares, that's 1 meter. Everything was opposite. So what I was trying to do was I was trying to push the selective landscape, right, in every type of ecological vector that I could identify. So it took a long time to identify where these ponds were. So what happened? So if there's a change, how much, and is it in the direction predicted for functionality? And so here's the Mare Lake, so counted the plates, etc., measured, looked at the rakers, a bunch of different issues like that. And so this is some work from a student, Stephen Lever, etc. I sampled those fish every year, and from 93 to 2008 generations, they breed every 2 years. And this is what happened. So over that generation, there was a 15% reduction in adult length. I don't know what the genetics of these are at this time. 46% reduction in lateral plates, 17% reduction. Yeah, I hear that. And so you go down, every one of these is going in the direction that you predict based upon functionality. None are going in the opposite direction, right? Which is odd because this is a bottleneck, a complete genetic bottleneck, and yet they went. Let's see. Okay, so it's called the phenotype, right? You don't know, so put through such a bottleneck, almost anything could happen, etc. So that was Steve, great student. He actually went into economics after this. He was lost in science. But my postdoc, David Marcus, and I'm sorry to say has passed away this year from cancer. He produced to me the finest work from my lab, etc. So I was able to collect enough fish that he was able to do whole genome sequencing, basically, right, of the transplant population and the source population, but also 28 allopathic populations that differed in their morphology, ecology, etc. And so what he found, which is what another colleague, Patrick Nozzle, was really expecting. Patrick Nozzle thought the selection would be really concentrated. But what David found was that there was evidence, FST value, right across all the chromosomes. It wasn't 1 or 2 markers, etc. So 62% mean selective coefficient, etc. And when you actually look at the targets, they actually may correspond to the QTL mapping of those different traits that change. So it wasn't just random genetic differences, it was those traits that, in fact, genetic traits that define those things that shifted in the direction. And shifted in the direction because when we looked at pond populations that are allopatric, we saw a lot of fixation in these different traits. Well, these exhibited the fixation in the same direction, right? So it was a pretty remarkable example, right? And so what can we say? I mean, close to finish here. Most of the genomic science differentiate, and are also the same sites that differentiate allopatric populations. And he summarized it this way. So the lateral plate, right, basically, if you take a look at the number of bony plates, right, and what he said, he basically said, let's compare how far did it go relative to these other populations. And so here we compared it relative to Mare Lake, the original source population, and then the transplant. So it was basically a very small shift in the numbers of plates, 6.6 to 6.4. But when we compare the number of plates in basically sort of lake population, it went 4% of the difference. So those, after 13 generations, went 4% of the difference, what we suspect 10,000 years ago. So that was for the numbers of plates, 18% basically for the streams. So when there's a lot of fish populations have a lake form and a stream form, and they're reproductively isolated. And so this is the distinction that these individuals, how much genetic difference it is. So when we go through the presence or two of lateral plate number 2, that's the second plate up there, what we got. So here was the difference between Mare Lake and the transplant population, and it's sort of modest, but then relative to lakes, it's 30%. It's gone 13 generations, it's gone basically 30%. How about streams? It's gone 50%. So what we thought was 10,000 years to produce those differences here is showing up in a relatively finite amount of time. So it's finite, and I won't go through each of these, but what you can see here is there's incredible high rates of morphological change. So he then did the genomic change, etc., with this. And obviously the overall nucleotide differences between, very, very little difference between the overall sites, etc. But when compared to ponds, there was about a 9% difference in the genomic sequence, etc. And compared to the streams, 20%. So in other words, the genomic differences were basically comparable to the phenotypic differences. Okay? And so when you actually look at relative differentiation, FSD, Mare roadside, the transplant population, you get a certain value. Now if we now look at, in fact, lakes, this is the difference between Mare Lake and another pond. We've got a 22% difference. So what happens now when you take a look? 41% difference. Right? That's how far the roadside, the transplant relative to differences between normal lake and allopatric stream or sympatric stream. So again, a high degree of differentiation over those 13 generations. And when you look at only at the outliers, obviously the ones that are targeted, being targeted by selection, compare those, it turns out that it's actually on average about 50 to 75% of the coding sequences of the genome are shifted in the direction that you predict. Not just a little part, right across the thing. Okay, so this is his summary. Many localized signatures of selection across the entire genome. Up to 70% of adaptive genome differentiation in 13 generations. Genomic change is driven by selection and is faster than phenotypic change. Essentially opposite to previous expectations, and the genomic constraints would hold back the potential for adaptation and coevolution. Because remember, this was a highly derived, basically, population of giant stickleback that had relatively limited genetic variability, and yet this is what it produced in a small number. So this is, I think, kudos to this unbelievable scholar, which I think is, if you were ever to read any of these papers, this is the one that I think is the one that stands out. Okay, finish off. Here's the lake, here's the adaptive landscape. Basically, well, I don't think of it anymore really as an adaptive landscape. I think it's more of it as a morphospace, basically. So we're looking here at characters, distributional characters, high, low, etc. Right? So this is not a fitness landscape. Everything I've told you and everything I've pretty well planned out, that this in fact represents the frequencies of niche available. And those individuals at the edge of the curve are not there who are low fitness have demonstrated they have high fitness good hair because there's not much niche space available to them. So this is entirely niche opportunity, nothing to do with fitness. There obviously is a fitness component, so all individuals are not equal. But when you look at these natural populations, etc., my conclusion here is that your fitness curve is pretty flat. Okay, are any of these processes and conclusions representative of other systems? I can tell you that almost all my colleagues hope not. Because it takes so much work to get what really should be basic ecology. Right, so anyway, that's where I am. Here's some colleagues. My best favorite here, Sheila, obviously Patrick Nozzle, etc. Dave Kingsley, Stephen Lieber, Sharon Bergstrom, Bruce Eagle, and David Marks. And of course, Andrew Henry and his offspring and students. Very good. Thank you.
I think hopefully everybody, the rapid evolution people, can see why we needed Tom to talk to us. Okay.